Hello, Maria.
Martina.
Hello.
Aiuto.
How are you feeling? How are you feeling, Maria?
A bit anxious.
This is the BBC from London. We'd like to interview you about your graduation, your forthcoming graduation. We'd like to ask you a few questions.
No, please.
How are you feeling? Are you feeling okay?
A bit anxious.
A bit anxious.
But also very emotional.
Is this your first degree?
Yeah, my first and also the last one.
And this is your sponsor?
This is not.
Yeah, my sponsor.
This is your sponsor?
I paid a lot for this degree. Yeah, really. But it's just an investment. I hope to give back some money. Give back money.
Okay, Charlie.
Okay,
This is a Monza. A Monza che si dice.
A posto.
Good.
Ma non sono per nulla ansioso, soltanto
No, per niente.
No, ti guardiamo noi, siamo noi che...
no, lo giuro, non so perché a me non faccio perché non devo studiare, si fermo su 10 anni.
Compagnia di studi? C'erano 3 persone prima di me. Sì, mi chiamano.
We're long to go.
Maria-Helena.
C'è da aspettare molto?
Altre due. Poi c'è la corona.
Grazie.
Kevin, are you here? Can you listen, please? Are you glad of the very last part at St. Mark's with Professor Kevin Hoffman, trying to understand why some patients develop colon cancer, some patients with Lynch syndrome develop colon cancer before some other with the same pathogenic variants, and one of the usual suspects could be the microbiota. Therefore, her thesis is on this topic. Do we have questions
it's
from
not
my
connected
hand?
yet.
No,
Not
not
yet.
yet. But we are in
We
time.
sent him the link and he said he was going to attend a congress, so I don't know if
Okay, we can go on.
Sorry, I'm sorry for
Okay, thank you, Professor Canestro. Today I would like to talk about the topic of my final dissertation, which is Lynch syndrome is associated with fecal and salivary dysbiosis. So Lynch syndrome is an autosomal dominant condition which is caused by defects in the mismatch repair genes and the EPCAM gene, and it is estimated that 1 out of 279 of the population carries a pathogenic variant in the mismatch repair genes. This syndrome increases the risk for colorectal cancer and other extracolonic tumors. In particular, Lynch syndrome-associated colorectal cancer is the most common cause of hereditary colorectal cancer, accounting for 3–5% of all colorectal cancer, and the risk to develop colorectal cancer depends on the pathogenic variant carried, with MLH1 and MS2 being the two most common. And the Lynch syndrome-associated colorectal cancer is because is stable. It is characterized by accelerated carcinogenesis. This is why colonoscopy is performed every 2 years according to the latest guidelines. It has an early onset between 44 and 61 and proximal location. As regards to dysbiosis, it is defined as an imbalance between beneficial and pro-inflammatory species. We have got dysbiosis which have been associated with different pathogenic conditions, both intestinal and extraintestinal. Among the intestinal, the microbiota alterations seem to play a role in sporadic colorectal cancer pathogenesis, with an increase particularly of Fusobacterium nucleatum, Bacteroides fragilis, and Escherichia coli. We also have oral dysbiosis, which has been mostly linked to periodontal disease, and the two most common bacteria associated to periodontal diseases are Fusobacterium nucleatum and Porphyromonas gingivalis, and both of them have been associated in previous studies to sporadic colorectal cancer and gastric cancer. And the role of microbiota in Lynch syndrome has been investigated by very few previous studies, with an increase of the following species, but the problem is that this study has some limitation. For example, they didn't compare Lynch syndrome patients with healthy control. Only one of this study compared 10 fecal samples of Lynch syndrome females with 8 healthy females, so all females, and the sample size was very small. In addition, they didn't take into consideration the oral microbiota. So the aim of this observational case control study is to characterize both the fecal and salivary microbial population of Lynch syndrome patients compared to healthy controls. We have compared 17 fecal samples from Lynch syndrome patients, 13 females and 4 males, with 21 fecal samples of healthy controls, 17 females and 4 males, as well as 37 salivary samples, 28 females and 9 males with Lynch syndrome, compared to 11 salivary samples of healthy controls. All Lynch syndrome patients were cancer-free at the time of sample collection, and the sample had been collected on average 9.2 years after previous surgery and chemotherapy. All the cohort was Caucasian, more than 18 years old, with a BMI less than 30, and exclusion criteria include other colorectal cancer syndrome, the use of antibiotics, non-steroidal anti-inflammatory drugs and steroids, GI diseases and metabolic disease, as well as alarming signs and symptoms like hematochezia and anemia, and a positive fecal occult blood test. Fecal and salivary samples have been collected with standard methods, and then we amplify the B3-B4 region of the 16S RNA gene, and the human and microbial reads are being discriminated using the BM tag. And the sample analysis has then been conducted with Kraken-2 for the taxonomic classification of the microbial reads, with Shannon and Simpson indexes to extrapolate the alpha diversity, the UMAP, that is an algorithm to visualize high-dimensional data, Pagan to calculate the relative abundance from Kraken-2 data, and dissect2 analysis to calculate the differential abundance. So before going on with the result, I would like to explain what alpha diversity and beta diversity are. Alpha diversity quantifies the diversity of microorganisms within a single microbial community, and we use the Shannon and Simpson indices to estimate the alpha diversity combining richness and evenness. So as we can see here from the example, in the community A, we have 3 species, the red, the blue, and the yellow one, and they are evenly distributed. So the Shannon index is high, while In the second community, in community B, we have a predominance of the red species, so it is pretty homogeneous and the Shannon index is low. The beta diversity instead quantifies the difference between samples from different groups from a qualitative and quantitative perspective. So here we can see two communities, two groups. In this case, both of these groups are similar because they have the same number of species, but in the first one we have a predominance of red and green one, so they are very different from the weighted point, since the second group is mostly formed by blue species. So we calculate the alpha diversity with Shannon and Simpson index, and the fecal alpha diversity was not different between Lynch syndrome patients and healthy controls, while the salivary alpha diversity was different between Lynch syndrome patients and healthy controls. We were not able to calculate the beta 1 because of the small sample of the small size of the sample. We also use the UMAP analysis to differentiate the fecal sample from the oral samples at order, family, and species level. And both in Lynch syndrome patients and healthy controls, the fecal and salivary samples are clearly distinct. We calculated the relative abundance with Pavian, and here we have a representation with the Corona chart of our results. So Lynch syndrome fecal sample have been characterized by decreased firmicutes, increased bacteroidetes, and increased protobacteria compared to healthy controls, while the salivary sample have been characterized by decreased protobacteria and increased firmicutes. Going more deeply, we have the fecal differential abundance was calculated by D-sector analysis, and the Lynch syndrome fecal sample are characterized by high expression of 79 species, high expression of 32 families, and high expression of 22 orders. Among the 79 species, the most relevant are 12 and mostly belong to the Bacteroidetes phylum. And here we can see that we also have Escherichia coli and Fusobacterium nucleatum that we have previously mentioned as involved in sporadic colorectal cancer. As regard to the salivary one, we have higher expression of 27 species, high expression of 2 families, and no higher expression of orders. Among these 27 species, we have a particular increase of Streptococcus and Bellonella species. These belong to the Firmicutes phylum, which is the most increased in salivary sample of each syndrome patient. We also have a slight increase of bacteria involved in periodontal disease for Pheromonas gingivalia and Fusobacterium nucleatum, but the p-value was not statistically significant. So to conclude, we can say that the Bacteroidetes and Protobacteria are the dominant phyla in fecal sample of Lynch syndrome patient. The fecal alpha diversity doesn't differentiate Lynch syndrome sample from healthy controls, and our studies suggest the role of gut pro-inflammatory bacteria in tumor development in patients with genetic predisposition, and similar findings to sporadic colorectal cancer. In addition, increase of fecal protobacteria enhances the permeability of the sterile inner layer of the intestinal mucosa, resulting in inflammation and closing continuum. While, as regard to the oral microbiota, we have that the Firmicutes is the dominant phyla of the protobacteria. We have the salivary alpha diversity that is different from Lynch syndrome and healthy controls, and a slight increase of bacteria commonly associated to periodontal disease has been observed in the Lynch syndrome sample, but since the cohort is small, we need a large cohort to establish if there is a link between periodontal disease, Lynch syndrome, and colorectal cancer. And thank you all for your attention.
Thank you, Maria-Helena. So we found your results pretty peculiar because of the differences from the oral microbiota from Lynch syndrome and normal controls. And I would like to ask you, before giving the floor to your supervisor, Dr. Puzzono, to ask you what will be the next steps in the research in the field of oral microbiota.
I think that the next step could be, for example, to compare Lynch syndrome patient with healthy control with a sample which is larger in size in order to establish if the p-value becomes higher, so if there is a risk to develop colorectal cancer in patients with Lynch syndrome and periodontal disease.
Okay, Marta?
Thank you for your work with us and for your thesis. You have told about the mechanism of action of the increased level of proteobacteria that enhance the inflammation carrying to colorectal cancer through the intestinal permeability. What is the mechanism of action conversely of the reduction of firmicutes? And do you think that this different mechanism of action could help in the future in the prevention of colorectal cancer?
Okay, thank you for your question. So I can say that firmicutes are reduced in our new syndrome fecal sample, and they are considered beneficial bacteria because they have a key role in the fermentation of dietary fibers which are otherwise indigestible by humans. So as a result of this fermentation, we have the production of short-chain fatty acids. Among these short-chain fatty acids, we have butyrate, and butyrate it has an important role in the intestine because it prevents inflammation, and it is also an energy source for the cells lining the colon. So if we have a reduction of firmicutes, patients are more prone to have inflammation. And I think that a possible role of firmicutes could be, for example, the administration of probiotics. So we can use these firmicutes as probiotics. In particular, a well-known probiotic is the Clostridium botulinum that in literature is already used as probiotic to maintain the intestinal health. And it also has an important role in suppressing the epithelial to mesenchymal transition and also the vasculogenic of sporadic colorectal cancer.
Thank you.
Thank you.
One more, should give it a point.
La smetta di parlare.
C'ho un firmicutes da regalarti. Brava.
Io vado, no,
Minchia di Kevin.
I had a drone. Many years ago.
la fosforilasi,
Ma chi sta a Milano? Appunto. Vabbè, ma se il tutto non è appunto. Non ci vuole la ricerca.
ne approfitto che mi hanno dato un microfono per dire due parole, le dico in italiano perché voi siete tutti italiani e grazie. Ho l'approvazione della signora alla quale ho dedicato in effetti il discorso. No, dico due parole perché questo è un momento molto importante nella vita delle persone. La propria giornata di laurea ce la ricorda sempre. Io la ricordo nitidamente, malgrado appunto l'età che passa, ed è una giornata molto importante. È un momento di transizione, cioè vi chiedo di concentrarvi un secondo sulla transizione, un po' come il parto, come diceva la dottoressa Poloniato prima, cioè è un momento in cui c'è un cambiamento. Questo è un momento di cambiamento, dovete fare secondo me mente locale al fatto che è un cambiamento. Da adesso in poi non siete più studenti, io vi chiedo un favore personale, basta studenti. Lo so che è una condizione di grande, come dire, benessere quella dello studente, perché siete abituati, l'avete vissuta per così tanti anni e la vostra specialità, come sarà fatta, tutti voi immagino entrerete in specialità, un pochino vi metterà alla prova su questo, perché sembra di essere ancora studenti, ma non siete più studenti. Dovete veramente pensare che da adesso in poi siete, fra pochi minuti dopo una meravigliosa proclamazione, siete dei professionisti, avete anche una laurea abilitante, quindi avete una laurea che vi dà pieni poteri di agire e dovete, come dire, essere esigenti su questo, anche con gli altri, non vi possono più guardare nello stesso modo, dovete chiedere. Uscite anche, malgrado tutto, con tutte le difficoltà, da un'università che ha cercato di formarvi e dovete essere fieri dell'università da cui venite, portarla spero nel mondo, poi magari entrerete tutti in specialità. Io sono sempre contenta quando entrano in specialità qua i nostri studenti, che io trovo siano superiori, almeno sono fatti come piacciono a noi, quindi ci piacciono sempre. Fieri dell'università da cui venite come siamo fieri noi di voi, e è un valore questo, vi dà una certa consapevolezza del vostro valore che non dovete permettere a nessuno di mettere in discussione mai, perché è una cosa reale. Siete bravi, siete preparati, siete secondo me il meglio che si può in questo momento. È terribile questo, Pietro, in effetti, e dire cavoli, io sono il meglio, però lo siete e vi chiedo veramente di averne piena consapevolezza. Insomma, se vedo meglio con senza gli occhiali. Okay, I declare that these graduates have fulfilled all the requirements for the master degree with the following comprehensive final grades. Graziana Grimaldi, 100 out of 110 with laude. Mariana Dovere, 110 out of 110 in laude with special honor. Pietro Valsecchi, 110 out of 110 cum laude. Maria-Helena Parrinello, 110 out of 110 cum laude. Emma Solinas, 110 out of 110. Beatrice Venerandi, 110 out of 110. As I introduced, by virtue of the authority vested in me by the Italian law, I confer the Master Degree in Medicine and Surgery a license to practice upon the following candidates: Graziana Grimaldi, it's very difficult, that's a more difficult part. We are a team. Pietro Bassecchi. Maria-Helena Parinello. Emma Sodina. Last but not Beatrice Venerale.
Possiamo vedere, può fare le foto. Sta qui dietro, tutti in alto.
Maria.
Lasci danza dei due. Mamma mia, tanta figlia.
Like 30 seconds to get ready.
What we're going to do?
Photo, official photo.
All of us?
Yes.
Where's Sally then?
Let me porta la capa.
She's over here.
Mettici il cappello.
La metto qui che fa più bene.
Mangia qualcosa.
Should have seen my mum's reaction when I told her she got jumping into a lorry. She exploded. crying her eyes out.
Alla fine è stata un'interessa anche di lei, della Maria-Helena.
We have two more, one for Sally.
Auguri Maria-Helena!
Avrei un dolorino qui, che può essere? Vivici in campo. Tutto il bicchiere?
Cioccolatino?
Mangiate, non li porto a Roma io,